All right, so it hits 1 and 01. So let's get started. So welcome everybody and welcome back. And I hope you had a nice winter break. And I would like to get started with today's lecture at our single-cell spatial analysis program monthly seminar series. To disclose my relationship with speaker today, Dr. Mario Suvà is my postdoc mentor. So I am so tremendously happy to be here and see him in this fashion. Thanks everybody for joining us today. And it is my absolute pleasure to welcome and introduce Dr. Mario Suvà, who really set the stage for the highest complexity of single-cell analytics at the genomic level. Dr. Suvà is an associate professor in the Department of Pathology at Mass General, Harvard Medical School, and Broad Institute of MIT and Harvard. He is also the director of the molecular pathology unit at Mass General. His laboratory is now leading the large-scale efforts in single-cell spatial profiling in clinical brain tumors. Dr. Suvà earned his MD and PhD degrees from the University of Lausanne in Switzerland. And in his background, we cannot see them, but his office has a coffee machine made by a Swiss company, Nestle, and also a huge framed photo with a beautiful view of Lake Geneva. So at the time, naturally, I sneak into his office and then enjoy drinking coffee with him and also without him as well. And that must be a wonderful place to do a PhD. And his doctoral research identified cancer stem cell in Ewing sarcoma. He then moved to the United States to work with Brad Bernstein at Mass General. He conducted postdoctoral work functionally identifying master regulatable stem cell programming in brain tumor. Lastly, I wanted to emphasize one of his impactful works. He is one of the pioneers of single-cell RNA sequencing in cancer. Historically, the first publication applying single-cell RNA sequencing to any cancer type was published in Science in 2013, which is his first publication as a corresponding author. This is now cited more than 4,700 times. And at that time, they obtained patient tumor, collected cells one by one, saw them physically into physically separated wells, and processed them manually. So they profile just only 400 cells, but its impact is huge by demonstrating the feasibility and utility of the use of the single-cell RNA sequencing. He literally opened the door for other people. Now, many people across the world use single-cell methodology to study cancer. Accordingly, Dr. Suvà received numerous awards. For example, the Merit Award from National Cancer Institute, the Merck Foundation Emerging Leader Award, Neuro-Oncology Scientific Award, American Academy of Neurology, and many, many more. So again, it is great pleasure to welcome Dr. Suvà for his seminar. Thanks, Mario, for joining us, please.
Thanks a lot, Toshi, for the very generous introduction, and it's great to see you, even though only virtually. I'm sorry I'm not there in person, but maybe in the future. Thanks for having me. All right, let me see if I can still manage to share my screen. Hopefully you guys see the slides okay. If not, just let me know.
We can see.
Yeah, all right. So thanks again for having me. As Toshi mentioned, I'm at Mass General, and I'm going to be discussing some of our lab efforts in the field of single-cell and spatial genomics in the analysis of brain tumor. So I have one disclosure, but it's not really relevant to today's talk. So I'm going to start by thanking all my collaborators. I mean, the reason why the lab has been doing well over the last many years is because I am fortunate to work with a lot of awesome people. And so I keep saying I frequently get too much credit for what I'm doing, but I'm really fortunate to have a lot of really amazing people I work with. The most critical over the last decade has been the lab of Itai Tirosh, with whom really we have a very strong partnership. His lab does a lot of the computational analysis on the genomics data that my lab generates, and we have been co-authoring papers together for a while. Of course, Toshi, who was in my lab, and now we still have a few ongoing collaborations, and many more than you can see on the screen. So I'm going to be talking about gliomas today, and I'm sure many of you are familiar with gliomas, but as a brief categorization of the disease, most of what we're going to be discussing today is related to this lower left panel here, so-called IDH wild-type glioblastoma, or primary glioblastoma in the old nomenclature. It's a disease that affects both children and adults, highly aggressive, and with very little therapeutic outlook. I'm afraid I think some people are not muted. If you can mute yourself, thank you. So glioblastoma is what we're going to be discussing mostly. I'm going to have some vignettes around some other entities, IDH1 mutant gliomas, that affect younger adults and are less aggressive forms of the disease, but nevertheless tend to be invariably fatal. So why do single-cell genomics and mostly spatial genomics that we're going to be discussing today? I want to just highlight a few of the things that we've learned over the years and how we're trying now to also learn them into the context of their positioning. So obviously, single-cell RNA sequencing has been very good at helping us delineating tumor cell lineages and stem cell programs, so we're better able to characterize the expression programs of malignant cells and relate them to developmental trajectories and understand which type of normal counterpart malignant cells are recapitulating. It allows us to really have a more granular definition of expression programs and developmental trajectories. It allows us to identify the cycling portion of the tumor and how those programs are related to other features like genetics or like developmental programs. My lab has been trying from the beginning, and we're getting better at it, to kind of concurrently capture both the expression but also the underlying genetics of each cell, so that we can place kind of this expression in the context of the underlying genetics. And we do that by either, like many others, reconstructing copy number aberrations from expression data, or also attempting to detect more granular either rearrangement of receptors or point mutations. And then that allows us to build phylogenetic trees based either on genetics, or more recently, we also do that based on epimutations. And it allows us to kind of order cells in their phylogenies and understand how these tumors evolve. But, at the difference of doing that from bulk DNA studies, we can understand at the same time what is the cellular phenotype and the expression program that is being evolved by these genetic events. And this really allows us to understand what is the cell that is being evolved, which is obviously the unit that matters in the disease. Over the years, we've gone back and forth between single-cell profiling, as many cells as we can, but it's historically not being that large of a number, even though more recently we've been able to expand. And so because what we learn tends to be limited by the number of samples that we've profiled, we've been using the single-cell signatures to reinterrogate bulk cohorts that are profiled at a much larger scale, so that we can better understand specific signatures in bulk profiles. And while my lab has been, mostly focused on cancer cells profiling, we've over the years also done quite a bit of work, and one of which was led by Toshi, on characterizing interaction between malignant cells and tumor microenvironment, and also cell-cell interaction, and just basic characterization of myeloid and T cells in these tumors. So maybe what my lab is the most known for is a paper that Toshi first authored, which is our effort to trying to break down the heterogeneity of glioblastoma into kind of like a comprehensible model. And I'm going to present a refined version of this today that will also include some of the spatial data, but the fundamentals have not changed. And so I'm just going to briefly go over this cartoon. I didn't want to go over the data because I've presented the story many times, and also we can just conceptualize what we've learned. But this is an effort in which we had performed single-cell RNA sequencing on glioblastoma. And by looking at recurrently occurring programs, what we had understood is that these tumors consistently contain the same type of elements. And these elements are different cell states that are reminiscent of neurodevelopmental cell types. And what we observed was that these tumors are consistently contain at least 4 subpopulation of malignant cells, and we named them according to what they best recapitulate as neuroprogenitor-like, NPC-like, oligoprogenitor-like, OPC-like, astrocytic-like, AC-like, and mesenchymal-like, which is not really a direct developmental cell state, but more something that relates to interaction with the immune system. And what we had learned in this effort is also that while each glioblastoma contains pretty much these elements, the relative proportion at which they contain them changes according to the underlying genetics, such that specific genetic events selectively expand certain compartment. And you can see here the relationship between CDK4 and NPC, PDGFRA and OPC, and EGFR and AC. And this really kind of provides us a model to describe the heterogeneity of the disease in the context of cellular composition, cell states, expression subtypes, and relationship between genetics and development. And it really, I think, has clarified a lot of the ongoing debate in the field. So in the more recent effort that also was co-led with Itai Tirosh, both studies are co-led by Itai Tirosh, we've tried to see if this model of heterogeneity could be resolved spatially, and what type of programs would we detect in glioblastoma if we did spatial genomic analysis. And so here, what was done was to take a cohort of about a dozen glioblastomas. There's a few IDH1 mutant glioma cases as well, but really what I'm going to be discussing mostly is the results in glioblastoma IDH wild type. And we used the prior version, what was at the time, of course, the current version, but is now the prior version of expression profiling by spots, which was the 10x Visium platform. And just for those of you, I know it's a spatial genomics group, so hopefully everybody's quite familiar with the technology, but just as a brief recap, these are spots that capture the expression of transcripts in defined areas on the slide. And these spots are, of course, spatially resolved, but are, of course, not cellularly resolved. And so they have a diameter of 55 micron, which is larger than a cell. Typically in glioblastoma, we've estimated it to be about 8 cells per spot. But it allows us to get expression spots in a spatially resolved manner. We complemented this type of approach with protein-based approach, codex, so this highly multiplexed antibody-based approach in which you can detect antibodies because they have kind of a fluorophore that's linked to a cross-linker that is easily cleavable. And we've done the antibody approach so that we can validate what we learn by expression, but also so that we have true cellular resolution, because of course antibody-based methods have true cellular resolution. And so it allows us to use 2 platforms for spatially resolving heterogeneity in these tumors. So the first step in trying to characterize the expression programs of spots by spatial genomics technologies is, of course, to try to first assign what major categories of cell types do you find in each one of those spots. And so, of course, in cancer, the first task is always to separate cancer cells from non-cancer cells, and we can expect that some spots will have a lot of cancer cells and other spots will have fewer cancer cells because, of course, we sample also kind of the adjacent brain. And so we do that by copy number aberration inference, which is really very similar to what we had developed for single-cell RNA sequencing. The idea is to look at the expression of transcript along sliding windows along the chromosomes. And if you see a lot of genes going up or down in their expression according to chromosome coordinates, you can kind of deduct that maybe there is a change in copy number of the genome in that region. And we know in glioblastoma this works very well in single-cell RNA sequencing, and we see here in spatial also it works pretty well. And we know it works well because we detect the expected copy number aberrations, such as chromosome 7 gain and chromosome 10 loss. These are signature for primary glioblastoma, and so we know we're doing a decent job. And then you can see how we can arrange spots that have good signal-to-noise ratio for copy number, presumably rich in malignant cells. And below here, you have spots that have very few malignant cells. Their copy number flat with this approach. And so we can see how we can classify spots as highly malignant, mostly non-malignant, and completely non-malignant. And it gives us kind of a first granularity of how to analyze these spots, because of course we want to analyze separately the malignant and the non-malignant spots, and also be able to interpret what's the underlying biology. So the second step is to ask, what each sample is made of. Now what we have the major categories. And to do that, we've really used an approach that, again, credit to Itai Tiroshi's team, is similar to what we've used when we did single cell analysis. So the challenge, as I'm sure many of you are familiar, is that if you take a handful of samples and you ask what's the difference between these samples, you will find a lot of effects that is sample specific, and that will dominate the heterogeneity that you're trying to make sense of. And because your ends are small, it will be very hard to make any meaningful conclusion. But here we approach the problem really on a per sample basis. And what we look at is the difference in expression between spots in the same sample. And we use different clustering algorithms that are better at resolving discrete patterns or more continuous patterns. And then we do that in a given sample. We see what the results are. And then we do that in a second sample, in a third sample, in a fourth sample. And then what we do is that we cluster kind of the conclusion. So we compare what are the recurrent patterns that we observe across samples and what is kind of the consensus picture that is emerging from this type of analysis. And this allows us to define, again, consensus metaprograms that consistently recur across samples. And that's kind of how we defined our first cellular states when we were doing single cell analysis, and the same approach applies here for spatial genomics data. So if we do that, we resolve spatial metaprograms, and the spatial metaprograms are depicted here. If it's bold, it means it's a malignant program. If it's non-bold, it means it's a non-malignant program. And you can see that the malignant cell programs that we recover are highly similar and concordant with what I described from the single cell studies. We detect an NPC-like malignant spot program. We detect an OPC-like, an AC-like, a mesenchymal-like. So these are very similar to the ones that we were capturing with single cell data. We have a few variations on the theme. We detect a hybrid mesenchymal associatic program. We detect a more proliferative metabolic state. And we have a state that is more related to the expression of a set of chromatin regulator that we will have to interpret. These are more unique to the spatial data. The other states or cell types that you see here are found both in single cell and in spatial data. So of course, one challenge is that the spots, I've told you, they're 55 microns in diameter. And so one could wonder if the programs that we detect here are actually programs that are really coming from single cells, or really could be a mixture or hybrid of different cell states combined. We're quite confident that these are unique cell programs because the approach, which is really, again, something that I was quite struck by, which is quite elegant. The idea, I'm going back one slide, but if you look at each one of those spots, let's say, even if they contain multiple cells in one spot, if you start comparing spot to spot, what will vary from one spot to the next is a certain frequency of a certain cell. And because the cells are the units that vary, if you analyze the expression between different spots and you do it across a lot of spots, you will end up picking up cellular programs, regardless of the fact that the spot is bigger than a cell. And so because you focus on the variation. And so we thought this would pick up pretty decently clean programs, and indeed it does. But to validate that's the case, we designed a panel of antibodies that are tailored to capture specific cell types or cell states of glioblastoma. And again, with this codex or Akoya phenocycler now type of approach, we were able to label all these slides and then repeat this type of analysis for clustering of expression programs based now on antibodies. And we can recover this by codex, the clustering of expression of the same cell states and metaprograms that I've just described by Visium. And so we're quite confident that these programs that we've recovered exist in the tissue, and they're not an artifact of the technology, and they're concordant both to the single cell and also with the antibody-based approach. So then comes kind of the spatial part of it. Now that you've kind of recovered the elements and you know what these tumors are composed of, you're really going to be able to begin to ask, how is this distributed in the tissue? And so to do that, we looked at spatial coherence, and so kind of how clustered versus unclustered these programs are in any given slide. And so you really can think of two scenarios. One, you have scattered programs in which spots that express a specific program are distant from the next spot that has this program.
Sorry, Mario.
Yes.
I think I am looking at somebody's screen by Peter Exma. I don't know why this happened. I'm not looking at your...
You're not seeing my screen anymore?
Yeah.
So you want me to stop and reshare again?
Yeah, but also like the Peter, can you stop sharing your... Can you hear or wait? Yeah. Sorry, could you reshare your screen, Mario?
Yeah, sure. Am I back on?
I think this is good. This is good.
Okay. Let me know if, thank you for letting me know. Let me know if anything else comes up. So we were at the kind of spatial distribution of this program. And so here, again, an example of either lowly spatially coherent programs with very scattered spots or highly spatially coherent programs with grouped type of spots. And so when we looked at the distribution of these programs, in spots across samples, we really see that in glioblastoma, we have mostly two scenarios. We have samples in which there is a fairly high spatial coherence with areas of the block and the slide that contain similar types of programs. You can see them here, for example, in yellow. And then we have other specimens in which really the spots are each distinct from each other with very little spatial coherence. So we call them either structured or disorganized. And quite rapidly, we observe that what tends to be the samples that contain high spatial coherence tend to be samples in which we detect the mesenchymal hypoxic flavor of malignant cells program, whereas the slides that are disorganized are slides that do not contain any type of hypoxic mesenchymal-like state. And so this would suggest that, mesenchymal and hypoxic programs kind of drive an organization of the tissue, whereas in areas that do not have this type of programs, we see mostly kind of disorganized spatial structure. We can, of course, do that. This is kind of a low-level assessment of what's going on, but you can be more systematic around assessing how this spatial coherence of programs is organized. And so you can, kind of analyze kind of spatial coherence across different sliding windows, looking at either what's, across up to 15 spots, let's say, from each other, or only looking at immediate adjacency of programs within just around a given spot, or you can look at co-localizations of expression within a spot, and you can just look systematically at how all these spots distribute and how these programs distribute. And so if we do that, we see that we recover a few expected associations. For example, again, linking back to Toshi's work, where myeloid cells and mesenchymal-like malignant cells are positively correlated. And you can see here also different flavors of similar programs that are positively correlated. Mesenchymal, basically mesenchymal and any immune cell types and or neurodevelopmental cell types to each other. So these are programs that tend to coexist across multiple scales within the same regions. But then we also have things that are anti-correlated, and this is mostly related to anything that's neurodevelopment with anything that's neural development. Those tend to be anti-correlated. And you can look at those associations one by one between each normal cell type and each malignant state, and do that also for immune cells. So if you do that systematically in the samples in which there is a high level of spatial coherence, so these structured samples. What you see is that in structured samples, you can actually resolve the large-scale architecture of these tumors into a model of 5 layers. And these layers organized in the following way. This looks like a cartoon, but it's a graph model. And the nodes and the edges are actually data-driven. And you can see how these 5 layers architecture of glioblastoma is a succession of hypoxic niche with mesenchymal hypoxic states, immediately adjacent, if you want, to the necrotic areas of the tumor, followed by a layer of reactive cells that still have a reactive component but are not hypoxic, and this would be mesenchymal state, inflammatory myeloid states, or this reactive mesenchymal astrocytic-like malignant state, followed by a layer of mostly immune cells and angiogenesis with vasculature, myeloid cells, and T and B cells. And then after that, we see the more neurodevelopmental related cell types that we've described, OPC, NPC, AC-like. And then this is kind of the infiltration of the normal brain parenchyma. And so you can see how this kind of spans the whole spectrum from the kind of the necrotic area of the tumor up to the infiltrative edge in the normal brain parenchyma. Now, I'm a neuropathologist, and to discover that necrosis organizes the tissue is not very, after all the efforts that we've put in, is a little bit disappointing. But the strength here is that this level of molecular organization goes far beyond what is visible by eye. By eye, you would know that this area is organized, but you wouldn't be able to resolve all these expression programs, how they're stratified. That's the first thing. The second is that we also saw that this type of architecture begins to appear in areas that begin to express hypoxic programs, but are not, frankly, recognizable as hypoxic by morphology. And so we think this is a more sensitive way to detect and also give us the molecular programs that are associated with this one of these states. This is just a summary of these figures shown with the codex data on top, in which you can see, again, spanning from the peri-necrotic hypoxic area up to the brain parenchyma infiltration with the codex. So this is the paper that, again, we published last year with the group of Itai Tirosh. And I'm going to show you now some of the follow-up that we're doing. The next few slides are more lab meeting style because it's some unpublished data that we're just beginning to look at, but is pretty exciting. And so I wanted to share with you guys. And then depending on time, I'm going to tell you some more upcoming work that is a more ready to publication, but I just wanted to stay in the spatial topic for you guys a little bit longer. So what are we doing now in terms of spatial analysis of glioblastoma? So we're leveraging the new technology. So rather than Visium regular, we're now using Visium HD. And so this is the latest version of the technology that has been developed. A few key differences. One is that this is FFP compatible, so we don't need, so the regular Visium was based on frozen samples. This can be done in formalin fixed paraffin embedded tissues, and so first advantage for a lab that studies a lot of primary human samples. Second is that the resolution is higher, so the capture spot here are now down to 2 microns rather than 55. You will see, it's actually annotated on the slide, that for the purpose of analysis, we tend to look at bins of 16 micron because they will contain more data, but we can break it down up to 2 micron in terms of resolution. And so this is an example of a glioblastoma. And just for annotation here, this is kind of the limit of the brain surface. Here is the pia, which is, the layer of the leptomeninges. And you can see here that the pia is destructed, and you can see here in the subarachnoid space, in the leptomeningeal space, that you have tumor infiltration. So this is an example of a glioblastoma that has broken kind of through the pia and has now invaded the subarachnoid space. So on the right is kind of the Visium HD analysis of this H&E slide, in which we're assessing the malignancy score. And the malignancy score is the same as what I've described before. It's mostly based on copy number aberration. And so we use the spots to infer how aberrant the copy number profile is. And then we can assign a certain level of confidence and a certain level of abundance of malignancy. And you can see how here in the subarachnoid space, we think there's a lot of malignant cells here in red. And you can see that this is mostly normal brain parenchyma, which kind of matches the H&E that you see on the left. But of course, we also have spots of tumor infiltration. You can see the vasculature is kind of preserved here, appearing as normal. Again, a lot of sanity checks to tell us that we're pretty good at separating malignant from non-malignant cells, which again is not a super high bar, but is reassuring to know that we're able to do that. So again, for some of the fun part now, because we have copy number profile that we can infer on a genome-wide scale, and we have this high resolution from the spatial data, we can now infer, if we say this is full of malignant cells, we're like, okay, so what's their copy number profile? And you can see here the copy number profile of these cells. They have gain of 7, they have loss of 10, so they're all glioblastoma cells. That's quite reassuring. But then you can see that they differ with other copy number profiles. I don't have, I don't need to go into all of the details here, but, loss of chromosome 8, loss of chromosome 22. These are different between different subclones. They're unique to specific subclones. And so it allows us to detect, how many subclones we can find. In this case, there are 4. And then we can see on the right how they're distributed in the tissue. And you can see here, subclone 4 is here, subclone 1 is here, subclone 2 and 3 are here. So which, first of all, gives us a first clonal idea of the distribution in the subarachnoid space for glioblastoma. And we can see also how the different subclones are almost independently infiltrating the brain parenchyma that is just below this subarachnoid space. And so it's really exciting for me to be able to begin to see that at this level of resolution. And we can, of course, because it's single cell expression data, we can also analyze what programs these cells express the most. And you can see here, mesenchymal versus radial glia-like program versus cilia-like program. Again, this is more lab meeting level. we haven't really narrowed down all the programs yet, but it's to give you a first glimpse of how we can begin to resolve the clonality in situ with really extremely powerful resolution in FFPE's embedded tissue. So, the sky's the limit, because now everything is like in a tissue bank in pathology is all FFPE. And so this is, I think, really exciting for the field. We can begin to look at, low resolution, but nevertheless exciting level clonality and how these things are organized. Just to focus a tad more just on the expression without the clonality, you can see here, again, just the architecture of this tumor, how it's really exquisitely represented at the molecular level. You see, for example, here the endothelial cell programs, how it's perfectly well lined up, and all the various malignant cell states that we can resolve. And I also show this to show that, we can detect around 2,000 count and around 1,300 genes for each one of those beans. And so again, it's a very high level of molecular information for each one of those bins. This is another example of expression. And I also thought this was, when I first saw it, I'm like, oh, we're finally seeing some light, because you can see the interferon gamma response, which is one of the programs that we detect, how it's spatially clustered and distributed in this tissue, and how it's bordering kind of the infiltrative edge of the tumor. So it makes sense that you're beginning to see kind of wave of response in the tissue to specific programs. I think this is, again, quite exciting. Because this is such high resolution now, we're also getting much better at detecting T-cell programs, which was something that was very hard to do with the regular Visium, because the cells are too small to really being captured with 55 micron spots. And you can see here kind of clusters of T cells in the subarachnoid space, and some of them inside the brain parenchyma. And to my satisfaction, I have to say, the computational biology is doing the analysis called these T cells, CD161 T cells, without me asking for it. And it's really satisfying because this is a program that a few years ago we've described with Kai Wucher-Phoenix team as being one of the T cells expressing programs that is relevant to gliomas. And we think it's a program that is an inhibitory pathway that can be modulated for enhancing T cell activation. But this was all based on single cell, a limited number of single cells that were not spatially resolvent. So to be able to see that in the tissue again with an orthogonal technology is quite exciting. And we'll see if, where that leads us, but it's been very interesting to see again. And this is just another example of this type of architecture that we can resolve and the type of kind of visually clustered already program that we detect in these samples. Just as a last vignette of where we're heading, also more lab meeting style than full presentation, we're also trying to generate data with this type of spatial technology. So this is the slide tags approach that the lab of Fei Chen has described, and I have an ongoing collaboration with Fei Chen and Dan Landau's team. And what we're trying to do here is to really get extremely good at genetics. We do that by performing in each single cell concurrent RNA sequencing and whole genome sequencing. And so each single cell is whole genome sequenced and RNA sequenced. We do that with the BioScribe kit and BioScribe technology. And we do that in frozen specimen in which we have used the slide tag technology that allows us to place spatial barcodes in the tissue. And so it allows us to capture individual cells for which we have spatial positionings thanks to UMI that are unique to their coordinates. And then we can recover the nuclei, and we can know their expression state, and we can know their underlying genetics thanks to whole genome sequencing. And so we're quite excited to be able to build high-resolution phylogenetic trees for these tumors in which we will have spatial information and also whole genome sequencing. Very much ongoing, but since this is a spatial group, I felt that I could share this type of early days data also for the sake of discussion. All right, so Toshi, maybe you can help me understand how we want to continue this. It was mentioned 45 minutes. I'm at 40 minutes pretty much, and I can either talk for another 40 minutes. I have plenty of other data, or I can take some question and answers now and then see how much time we have and see if I should go on with another story. What do you think?
Yeah. Can you go maybe like 10 minutes-ish for the another talks? Yeah.
Okay, 10 minutes.
Yeah.
So 10 minutes, let me try to do this in 10 minutes, I'll manage. Okay, so here is not spatial, and that's why I apologize to you guys because I know the topic is spatial, but here it's still glioblastoma, and now you guys are all familiar with the states. We've tried to analyze how these states change over time because, most of our studies have been done with a single snapshot of expression programs of glioblastoma. And what we're trying to do now is capturing the expression state in time point 1 and see how that evolves in time point 2. And so really trying to understand what is therapy doing to these tumors. And we're doing that in the context of standard of care. But more importantly, I think we should, as a field, we should be doing that in a lot of experimental therapies for glioblastoma because we're consistently not really doing a great job at curing patients, but maybe with this technology, we can get better at understanding if we're doing anything at all and how we're moving the needle. And so really the question here is like, this architecture of 4 states that we've described, how does that change at recurrence? And so to do that, we've created what we call the CARE Consortium for cellular analysis of resistant evolution. It involves my lab, in my lab, Masashi Nomura, Itai Terosh's lab, and Avishai Spitzer in his lab, the lab of Antonio Iavarone, and the lab of Royal Verhaak. And we got samples from across the US and actually from across the world. You can see Tokyo, Seoul, Norlux, Paris, and Canada also represented here. And the cohort that we have is 121 specimen, 60 pairs basically, all IDH wild-type glioblastomas sampled at different time points with very rich clinical annotation. So this is a data set of about 500,000 single cells. After QC, we profiled almost 1 million single cells, but half of them failed our QC. So that's just what it is with the primary human samples. You have to deal with not all the samples would be of great quality. And so you can see here, kind of the high-level view of what these tumors are made of. We see the expected subset of cells, malignant cells, myeloid cells, oligodendrocytes, neurons, and other cell types. The value here is that given the size of the data set, we actually get a pretty good representation of the fraction of cells that we get across all these samples. And so the numbers that we detect are, relatively informative in terms of what's the relative abundance of this cell type in these tumors. And you can see, for example, for T cell, it's down to 1% of the tumor. Malignant cells, it's about 2/3, but you have to deal with a lot of myeloid cells in the microenvironment. So we've tried to break down, and again, I'm going to do this short because we don't have a lot of time, but try to give you the high-level take-home message. So again, we always try to first classify the samples based on how much malignancy they contain. So we will have samples with high malignant cell fraction. Most of the cells in that sample will be malignant cells. Intermediate, half and half malignant and non-malignant cells. And then low malignant fraction with a lot of tumor microenvironment and immune cells. And of course, subcategorized based on the dominant non-malignant cell type. So the first layer that we see is that, well, if we look at all these samples, we can classify them according to how many malignant cells each one contains. So that's the first layer of heterogeneity that we see between samples, is cellular composition. The second layer is, what did each one of those samples contain? And so it's our malignant cell states. So we repeated the same type of analysis I've been describing now for a while, which is, the variation of malignant cells within each sample, and which type of metaprograms do we detect. And for the sake of time, I'm just going to tell you that we detect kind of the expected suspect with maybe a little bit more granularity because we have much higher numbers of cells and samples. And so rather than 4 states, we detect 9 states, but they're all tend to be subsets of stuff that we had already described. And then the third layer of heterogeneity that we've been able to describe with this type of approach is what we call baseline programs. Basically, what we did here is to say, if you compare sample 1 to sample 2 and you ask what's different, most of the time the answer will be, well, they're different either because they contain different cell states of malignant cells, or they're different because they contain a different fraction of malignant cells. So these first 2 layers explain a lot of the heterogeneity. But if we go beyond that, we can actually begin to compare malignant cells in the same state and at the same level of abundance between different samples. Will they express the same programs? And so you can compare, it's the cartoon here on the left, OPC to OPC, let's say AC to AC. So it's matched for cell state. It's matched for composition because we make mini pseudobulks that contain the same amount of malignant cells. And then we ask, if we do that, do we resolve every heterogeneity in glioblastoma? The answer is no. There's still heterogeneity in baseline expression programs, and some samples will express a specific program that we call the ECM program, other samples a neuronal program, and yet other samples a glial program. And this is really what's left after accounting for cell state and for composition of malignancy. And so we think this is kind of the baseline heterogeneity that glioblastoma has. To kind of circle back to the spatial, if we ask, between the three layers, malignancy, cell states, and baseline program, are these feature independent? We actually see that they're not, and that these features tend to covary, such that, for example, tumors that have an ECM-like baseline programs are the same tumors that contain a lot of mesenchymal-like and hypoxic-like malignant cells and a lot of immune cells. Tumors that have a neuronal baseline program are those that contain a lot of non-malignant cells of the brain parenchyma, and the AC-like and GC-like cells are the samples that have high malignant fraction. And so If you want, the immediate microenvironment also influences the baseline program of these cancer cells, and the features that I've described kind of in isolation are actually associated in a high-level architecture, and these architectures tend to also be associated with specific genetic events that are described here. And again, it allows us to mostly take our understanding of glioblastoma heterogeneity to a layer in which the features that we tend to describe in isolation can be interrelated. And so it allows us to define what we call ecosystems of, either glial, ECM, or neuronal that varies both in terms of abundancy of malignant cells and type and states of malignant cells and baseline expression profile that we think can allow us to explain heterogeneity in glioblastoma really to a more granular level. To finish the loop of this story, I'm just going to say three more things, which is that this type of result can be obtained when you analyze a large cohort of glioblastoma with these technologies independently of time point. And so this kind of ecosystem that are described here are found across all the glioblastoma, and this analysis is robust independently of time of sampling. And so then we ask, well, if it's independent of time of sampling, is there anything that changes, or are these ecosystems affected at all by therapy, which is kind of what we set out to do in the first place, assess how they change over time and after therapy. And so here now we tend to reanalyze the data, but we fixed all the primaries and all the recurrent samples in their respective categories, and we ask what varies now that we've described kind of the baseline architecture of these tumors. And so if we do that, we see that really the one thing that changes the most is the first layer that I've described, cell type composition and malignancy fraction. And so you see the primary samples have a higher fraction of malignant cells here in red. The recurrent samples have a much lower fraction of malignant cells. This is consistently seen and consistently found across all our cohort. So lower tumor purity at recurrence, higher fraction of kind of a glioneuronal microenvironment at recurrence. And that's pretty much it to make it very fast. Malignant cell states doesn't change much at recurrence. Baseline programs doesn't change much at recurrence. There are some small trends, but there really aren't robust statistical data to support that. So we really think the one thing that changes is the composition of the tumor in terms of malignant cell fraction, not so much the other layers of heterogeneity. And the associations between the layers that I've described is conserved in primary and recurrent time points, these different ecosystems, their features or their co-segregation of features. You can see how the clustering of the features is preserved in primary and recurrent time points and is independent of the time of sampling. Just to tell you that it's not completely equivalent, the before and after, the nice thing about these data sets is that we have very rich clinical annotations. And so if you start digging into specific subsets of tumors, so if you do a global comparison, the story is what I've told you, there aren't that many differences. But if you start to subcategorize and look at specific subsets of tumors, you're beginning to see some effects. And so, for example, this is one subset of tumors in which we see the so-called small deletion phenotype. This is a radiation-induced genetic scar that is detectable on the genome when you do whole genome sequencing or whole exome sequencing. And we see that those samples that have this genetic scar from radiation are enriched at recurrence for hypoxic-like malignant states. That's one first association that we see. We also see that samples vary in their trajectory according to the MGMT promoter methylation status. This is one of the most robust clinical stratifiers that is used to assess what the response to therapy will be to glioblastoma, with the unmethylated patients doing far worse than the methylated patients, that because the tumors respond less well to alkylating agents therapy if you're unmethylated. And we see that the trajectory varies accordingly. And this is depicted here with an enrichment for mesenchymal-like hypoxia in unmethylated, and a reduced representation of the same program in methylated patients that presumably respond better. And so, again, this is my last slide so that I'm going to be on time for the discussion. The idea is that after you sequence a lot of single cells in glioblastoma at primary and recurrence, you really only see one clear effect, reduced malignant cell fraction. The rest is a lot of noise that is patient-specific, and we're excited to report that because the field has been excited about other trajectories for a long time, but we think that's confounded by bulk analysis. And then when you do single cell analysis, the picture is not as clear as was thought, and we only see predictable trajectories when we have clear modifiers of clinical response that we can identify. And that's it. Let's stop here. I'm just going to thank everyone and take your questions. So this is the lab, more recent version, and you can see the alumni well represented with Toshiro Hara high in position there. And thank you for your attention, and thanks. Happy to take questions.
Awesome. Yeah, thank you so much, Mario, for the fantastic talk on the application of multiple spatial approach and also massive single-cell RNA sequencing longitudinal studies, data-driven way to understand the disease, and also introducing system biology to understand the building block of cancer. So yeah, we have time for a couple of questions. If you have questions, please unmute yourself or raising your hands using Zoom function so that I can navigate to you. So any questions? And I think that June has a questions, please.
Hi, Mario, nice talk. I want to ask about the relationship between the cancer clones 1, 2, 3, 4 and the tumor state, and how they correlate with the classic types, proneural mesenchymal, and so on and so forth.
Yeah, great question. So the clones that I've described, 1, 2, 3, 4, again, this is more lab meeting level analysis. So as of now, I don't think they have any relationship with the states described or the subtypes. This is just an example of a tumor with 4 subclones. Actually, when we've done this in the past, if you look at the expression of subclones in glioblastoma and in other cancer types, that differ by copy number aberration, the difference in their expression can be explained at 99% by the copy numbers differences that they harbor. So, the genes that will be differentially expressed are genes that are sitting on the chromosomes that are differentially present. And so most of these subclones differ in expression on genes that sit on the chromosomes that are different.
So the clones have a inferred lineage relationship. Are those tumor biological theme or the states? Do you also have a before and after relationship?
So I'm not sure I fully understand. So they have a before and after relationship in the sense that we can kind of order them to some extent.
Yeah, in your state map, there are like 4 blades of a flower.
Yeah.
Do you also have a sense that some state grow out of one of those blades?
Yeah, so no, we do not have that sense. There's not a direct relationship between the state maps and the phylogenetics. And actually, even when we do much higher resolution phylogenetics and we look at subclones, we see that those programs are present in in a lot of the subclones. And it's almost how, by definition, we found them. They're recurrent, and they're concurrently found in many subclone branches of the tree. The exception is that copy number events that affect specific drivers will have an effect on the expression programs. That was our description in the original paper. So CDK4, PDGFRA, EGFR, those affect those expression states. But most of the other copy number events don't affect this.
Thank you.
Thank you. Sriram, so great to see you.
Same here, Mario. Really fantastic talk. Really, really awesome. I think the data is really awesome. So it's sort of counterintuitive that the recurrence has lower number of malignant cells, right? Do you think this could be an artifact of how recurrent surgeries are usually done? Maybe they just take a little biopsy, or they don't do like a whole resection at the primary time. Yeah. So what are your thoughts on that?
Yeah, no, I think it's possible that there is some surgical bias in what we're getting at recurrence. However, I do think it almost makes sense that at recurrence, you will have a more infiltrative tumor, because that's kind of like what's left behind after the first mega cavity resection. And what you end up re-resecting is something that's more deeply infiltrated into the brain parenchyma. And because of that, you end up with lower malignant cell fraction. You maybe don't have such a cancer cell dense primary mass. So I guess to some extent, that's our explanation.
Yeah.
But of course, yeah, we're always limited by what we get.
Yeah.
And
it's also really striking that the components don't really change, right, between the primary and recurrence, I guess the architecture or the tumor subtypes. Do you see any like profound changes in the immune-related signatures?
So we see some trends, higher kind of macrophages over microglia type of immune infiltrated recurrence, but they don't dominate. What dominates is the higher fraction of neuronal microenvironment at recurrence associated with this low malignant cell fraction. And I think, I'm actually glad that, Royal is a co-author on this paper, because, we've been struggling with how do we reconcile this with this long-standing literature around mesenchymal being enriched at recurrence. But I really think it's mostly driven by specific clinical subsets, and also confounded by the bulk analysis. We really can't see that trend as a global trend. We only see that trend in specific subsets of clinical subsets of the disease.
Awesome. What a fantastic talk. Thank you again.
Great to see you, Sriram. Thank you.
Here.
Awesome. And also we see some comments in the chat. I see 2 questions from Megan and Taiwa. So Megan, can you unmute and ask your questions? Or I can read your questions.
Yeah, here, sorry about that. I'm in a communal office, so it might be a little bit noisy. I was wondering if you found whether the ecosystems or the baseline profiles predict patient prognosis at all.
Yeah, good question. So no, we did not find that. And I think it's, because to some extent, once you stratify your cohorts robustly, all these group of disease tend to behave pretty poorly. And so the level of stratification that you get is limited. What we rediscovered is what is already known, which is the level of expression of MGMT, which is this gene that is involved in the response to the treatment, affects the time to recurrence and the surgical interval. And that's also, again, we make a point in the paper around being able to quantify that directly from the expression of the genes in the single cell data, which is something that might be of interest because the field has struggled on how to correctly quantify this expression. But anyhow, we get basically the same clinical modifier that is already known, but nothing new with this type of analysis.
Thank you. So one question from me, actually. So you mentioned about the prognosis and also talk about the mesenchymal program. So in general, in the field, people think the mesenchymal is very bad. Early on, the mesenchymal subtype is like the poor prognosis. And still in Lloyd-Warhacker's paper, it's associated with some like the shorter recurrence-free survival, maybe a little bit, but there is not that much differences. So what do you think about, and also like in your spatial analysis, like a mesenchymal tumor is like at the core, not at the periphery. So do you discuss a little bit, for example,
I mean, that's a good question. I mean, I don't know exactly what to make of this. The effect on survival of the subtypes has always been pretty modest. It was stronger before people recognized IDH completely. So once you remove the IDH, I mean, there's still some, a little bit of an effect, as you say, in terms of like strength to response to therapy, but it's a subtle effect. I don't know what to make of it. Like, I think, it depends a little bit on the cohort composition. In our cohort, which is still a pretty sizable one, we don't see that effect. So I think mesenchymal is just as bad as any other of those states, I think is the message. All these states are bad, and I think the relevance of the subtypes and the states is mostly to me to think about their elimination in a different way. You probably need a different strategy to eliminate the mesenchymal than you need to eliminate the NPC, OPC. But that's mostly biology rather than clinical outcome at the moment.
Agree. So for the questions, we still have 2 more questions in the chat box. Dr. Chun has some questions. Could you unmute yourself and ask your question?
Hello, great talk. So I was wondering about early event of tumor genesis. So if you have multiple samples, then small and large, hypoxic and not hypoxic, can you infer that speculate what's really happening in the early stage of tumor genesis of glioblastoma, even before I know that at the time of diagnosis, tumor is pretty big. But from your database, can you infer that time course of that tumor development?
Yeah, that's a great question. You know, I've been hoping to be able to do that for a long time, also in our efforts of low-grade gliomas that are, less, formed at the, or like, less aggressive at the time of sampling. But we haven't really been able to do that yet. I'm hopeful that with the spatial technologies and also with this whole genome sequencing that we're doing, we'll be able to track back the history of the tumor as much as possible. But you have to always remember that, as Sriram reminded us, we're only analyzing what we're getting. And these samples are actually quite big, and our capacity to comprehensively analyze a tumor remains very limited. And we don't have really good access to whatever would be an early glioblastoma. It's always a glioblastoma that's clinically manifest. And so it's very hard to do the early lesion. I think the best chance of doing early lesions are mouse modeling. And, there's a recent paper from Peter Dirk's group that looks at that. But early lesion in humans is difficult for glioblastoma.
Thank you. And given the interest of time, I think the next one is the last questions. So Paraba, can you ask a question?
Thank you. Great talk. So my question was about the clonal composition, like you showed in one Museum HD section, that there are 4 subtypes of 4 subclones within the tumor within one patient. I was wondering 2 questions there, whether the clonality, like what additional chromosomal aberrations that are acquired by the cells is in any way related to the vicinity, like the other non-malignant cells in the vicinity. For example, those that are close to macrophages have this particular type of clonal evolution or not. The second question is whether the type of clone has any say on how the patients do, on their survival or their recurrence.
Yeah, thank you. Good question, but unfortunately, we don't have enough data on either of those questions yet. So the subclones that I've shown you, the 4 subclones in this sample, is just whatever is happening in that sample. I don't know how representative it will be of a larger cohort. We've currently analyzed like 3-4 samples by Visium HD, so not enough to draw any conclusions. I expect to detect a few subclones in each one of them, but the subclones that we detect tend to be very much patient-specific. Then there's no recurrent pattern of subclonality that we can see across samples. And so it tends to be more specific to that whatever patient sample. And because of that, we also don't really have associations with microenvironmental features or outcome that we can comment on. Too early. Maybe a year from now, I'll have a better answer. Thank you.
Awesome. So yeah, thank you so much, Mario. I think all asked the questions, and it is good time to close today's seminar. Thanks, Mario, for your fantastic and inspiring talk. Thanks, everybody, for your attendance. So happy Monday, and thank you.
Happy Monday. Thank you so much for having me, Toshi.
Thank you, Mario.
Thank you. Thank you, everyone.
Thank you.
Thanks, everyone. Have a great week.